Yeah, hello everybody. I'm sorry that I cannot be there in person. Um, so it has been a long and stony road to develop therapeutic approaches for Alzheimer's disease. Took more than 100 years, but we had a couple of symptomatic drugs already on the clinic, and now there have been some really exciting developments with the antibodies, so the MAMAP being the latest to be approved. But at the same time, we all agree that that's not the end of the road. Alzheimer's disease is a multifactorial disease, and the different phases are characterized by distinct pathologies. So certainly A beta pathology is an early event, but there are other issues actually that affect many different cell types in the brain, such as microglia, astrocyte, the neurons that eventually lead to the cellular changes that cause cognitive decline. So this is why we also need additional therapeutic approaches, and this is what the field is also doing. So there are a lot of clinical trials going on, basically, that address pathways in addition to amyloid and tau to reflect the different stages of AD, also the stages when causative therapies are likely to fail. And this is actually a very important thing because effective therapies have to be early. So the time point when Alzheimer's is diagnosed, basically, the pathology, the molecular pathology is already much advanced, and causative therapies would not work anymore in a brain that is also already suffering from massive neuronal loss and degeneration. So we know that there's a time window where therapies and prevention even might work, but this is long before cognitive decline occurs. So how do we detect these individuals that would benefit from such a therapeutic strategy? Well, there are markers already that you can measure in the CSF or using MRT, but these are time-consuming, invasive, and also expensive. So what we are looking for is biomarkers that are minimal invasive, cheap, and can be used for screening approaches. And our answer to this is this. Don't worry, Zoom is still working. What you see here is the dark matter of the genome. And the cool thing is that only 1.5% of our genome codes for proteins. The rest of the transcriptome is non-coding RNAs, and these have been considered to be the dark matter of the genome. So there are small non-coding RNAs, there are long non-coding RNAs, and they really open up a lot of novel space for biomarker development and therapeutic interventions. So today I'd like to talk about microRNAs in this context. MicroRNAs are produced in the nucleus, they are processed, they are short nucleotides, 21 base pairs long, and then they are loaded basically on the RISC complex, the RNA silencing complex. And with this, they scan mRNAs, they bind these mRNAs and then either block translations, so they regulate proteostasis, or lead to the degradation of the mRNA by this regulating gene expression control. And why is this interesting in the context of Alzheimer's disease? Well, we all know that Alzheimer's disease is affecting many cellular pathways. So there's always beta amyloid tau deregulation. There's always neuroinflammation. There's always deregulation of synaptic plasticity. And some proteins go down, some mRNAs go down, some others go up. So let's look at this individual. So let's say this guy has a certain protein that is deregulated in this pathway, 2 in this, and 1 in the plasticity. And this person, if you measure these proteins, you can say, yes, it's on its way to develop AD. Now, the person might have a very similar phenotype, very similar pathways deregulated, but it's slightly different proteins. So with the same biomarker panel, you would not pick up this guy here. So the solution to this, or in addition to these other measurements, we think that microRNAs might be an interesting option because, as I mentioned, one microRNA can regulate many mRNAs at the same time and many proteins. They often regulate entire pathways. So by measuring one microRNA, you might learn a lot about a specific pathway linked to these features of AD here that are used in this example. And at the same time, you also amplify the system because you measure the response of many different proteins. Now, microRNAs can also be detected in blood, and they actually interact across organs, so they mediate inter-organ signaling. So they come from the brain, and you can detect brain-derived microRNAs in the blood. So the story that I want to tell you today is a paper that has just been accepted, and that really was a group effort with all the people that you see here. So what we did was we provide here the first small RNA sequencing data on the ADNI cohort. And we had really well characterized and selected patients that were classified as control, early MCI, late MCI, or Alzheimer's disease with follow-up phenotypic data. And what we wanted to do in the first approach is to create the microRNAs from machine learning and then identify microRNAs that can predict the diagnosis and the conversion from MCI to AD. And The overall aim was to find microRNAs that can aid cognitive screening approaches that you can do in a primary care setting to identify people at risk that could then go into follow-up diagnostics. Just some quality control. So we had a lot of control samples that we sequenced across all the batches. These were 21. And what you see here, it's red. So red means there's a very strong correlation between the samples across all the batches. So that means the data is really good and not that much affected by technical confounders. Well, there were some samples that were not as good as you see here with some other quality controls. So we removed the 44 samples and then had in the end 803 samples that we could analyze. Now, we detected 300 microRNAs in plasma, and we wanted to put this into machine learning. So in principle, we could look at 2 to the 300 combinations. Now, that is more than you have particles in the universe, so we had to boil this down. And we did this by looking first at microRNAs that would correlate with a diagnosis, or with a conversion of early and late MCI, did some initial machine learning, and then came up with 73 unique microRNAs that we put into our machine learning classifiers. Now, the first thing we found was a signature that was predicting AD, and that was having an AOC of 0.72. So this is a ROC curve analysis, and the more this curve here goes to the left, the better the biomarker is. So ideally, you want to have 0.1. 0.5 is random. So this is actually okay, but it's not good because all the CSF markers and even the mini-mental state is much better. But that's not a problem because we don't want a marker that helps us to detect Alzheimer's disease because this we can do very well. We want to have markers that help us to identify at-risk patients. So let's look at early MCI. We found another microRNA signature with an AOC of 0.71, which may not seem good, but it's better than the CSF biomarkers that we have. The same for the late MCI. We have an AOC that is, decent, but cannot be used really in the praxis. But it's as good as the CSF markers, which are, of course, much more invasive. Now, when we combine now certain microRNA signatures, even with the cognitive screening tools like the mini-mental state examination, you see that you get up to an AOC for detecting late MCI, for example, of 0.75. And this is as good, if not better, than this more invasive CSF biomarkers. Now, another very important and probably more important thing is the conversion from early MCI to late MCI. So we had follow-up data. This is just a demographic. So there were quite a few people that had stable early or late MCI, and then there was a huge group that converted within 24 months. That's the data that we are looking at here now. And there we find another microRNA signature that could help us to predict if somebody would convert with an AOC of 0.7. And this is better than the CSF biomarkers, at least in the group of patients that we have analyzed here. For the late MCI, it looks even better. So we have an AOC of 0.75 with this microRNA signature, which is really much better than any of the CSF biomarkers, giving us hope that we can build on this data to develop, hopefully, some screens that can complement other approaches to diagnose people who will convert. With the mini-mental state test combination, we didn't find anything here. It's not a good test for MCI, of course. But if we combine, for example, the microRNA signatures with other cognitive screening tools that are a little bit more complicated, like the ADAS-Cog, we can get the AOC, at least in the late MCI, up to 0.79. So that's already, I think, quite promising. Now, this is a summary. So we find signatures for AD, early MCI, late MCI, and for the converters. And there are some microRNAs that we find, for example, 142 is an AD. It's also a part of the signature that predicts conversion, but most of them are really distinct. And this is important because initially I said that Alzheimer's disease is characterized by very distinct molecular phases. So we believe that we can develop microRNA signatures that help us to capture these distinct phases and then really have a customized, personalized treatment for these individuals at a certain stage. The other important thing is that there have been many studies before that looked at much smaller cohorts, looking at microRNAs in plasma, blood, whole blood, or serum, for example. And all of these microRNAs that we identified have been, in one way or the other, already shown in the literature to be deregulated in AD or MCI. So this is a nice confirmation of the quality of our data. Now, microRNAs regulate mRNAs and proteins. So you can look at their targets and ask what kind of pathways do they reflect. That's what we did. And when you look first at the diagnosis, early MCI, late MCI, and AD signatures, different microRNAs, If you come from the AD, you see the top 15. There are pathways coming up like Alzheimer's disease, long-term depression, HIF-1 alpha signaling, and most of these you also see in the early MCI and late MCI. But interesting are the ones that are specific to early MCI. And here you have ferroptosis, cannabinoid signaling, oxidative phosphorylation, which have been associated with early AD pathogenesis. And for the late MCI, we have more pathways that are specific. This is reflecting inflammation and, for example, cholinergic synapse deregulation. Also makes a lot of sense. We also looked at this basically from the converter point of view. So we have a couple of pathways that are similar actually in the early and late MCI converters. But what's very interesting is that the people who have early MCI and convert to AD within 24 months, they have pathways that you usually only see at late AD. So mitophagy, synaptic vesicle recycling, long-term potentiation is already deregulated here, suggesting that they are really on a much faster path to develop AD, whereas the late MCI converters have mainly metabolic and inflammatory pathways that are specific to this phase. Now, to sum this up, we believe that the analysis of the blood microRNAome can really aid screening approaches to identify at-risk patients for Alzheimer's disease. And the other thing is that specific blood microRNA signatures likely represent distinct molecular phases of AD pathogenesis, which is very important if you want to decide which therapy you want to give to a certain patient. Now, this is far from being complete, so there's a long way until we see this basically into clinical routine. So the first thing that we do now, we have a replication cohort, the DZNE Delcode study, which we already sequenced, and the data looks good. The analysis is currently going on. We are doing the same actually in individuals from the Framingham Heart Study, where we have not only plasma, but we also have postmortem brains from the same individuals. And what eventually we need to do is to transfer this microRNA detection from sequencing approaches, which are still quite complicated, to easier assay formats. With this, I come to the end, and I really like to thank all the people that contributed to this, the funding agencies, and of course, our collaborators that work all together on an NIH grant to identify microRNA signatures that aid the diagnosis of Alzheimer's disease. Thank you very much.